Let's talk about carbon reactions. So I'm going to have a short video on the carbon reactions that are part of photosynthesis, and then I'll have a second short video for this week on the supply of substrate to these carbon reactions. So carbon reactions are taking that energy that was harnessed through the light reactions and using that to assimilate new carbon into plant tissue. So again, the main products from the light reactions that we talked about were ATP, NADPH, and NADH. And so those molecules are then going to go into kind of supporting and providing the energy to drive these carbon reactions. And as it incorporates carbon into the tissue, this does a couple things. It provides carbon skeletons that can be used for building, bigger tissues. So, and then it also provides a longer term storage for energy, because it can take these compounds it's going to make through the carbon reactions and then break them back down later or somewhere else in the plant and release the energy that's stored in those chemical bonds. So those are two of the important parts of two important ways the products of the carbon reactions are used within the plant. So let's talk about the carbon reactions, and all of the general reactions we're going to talk about are involved in the Calvin cycle. And we're not going to talk about the detailed steps, but just kind of the general processes that are important in the Calvin cycle. So the Calvin cycle is the process in a series of reactions that allows plants to incorporate new carbon into their tissue. And they're taking that carbon from CO2 molecules that diffuse into the leaf and end up at the point of carboxylation. We'll talk about that more in a second. And so we'll talk about the location that this is happening, and then we'll talk about the general steps and products of the Calvin cycle. So location, let's go back to these figures. So I showed these figures during the light reactions. And so on the left here, we've got the plant cell. Remember, in a leaf, you've got the layer of cells at the upper portion of the leaf. That's the palisade layer. And remember that our chloroplasts are generally concentrated there. They can be in other cells as well, but that's generally where they're concentrated. So we can kind of think about these reactions happening in that upper portion of the cell, the upper portion of the leaf. And then remember, all of these reactions are happening inside the chloroplasts. And so the picture on the right, we also looked at last week. This is just a cartoon of a chloroplast. Remember, the important thing about the location of the light reactions is that all of the enzymes involved in the light reactions are embedded within the thylakoid membrane. So remember, this kind of light green here represents the thylakoid membrane. And then it creates these stacks. They're called thylakoid stacks. Or all of these membranes kind of stacked on top of each other. And the enzymes are embedded in that particular membrane. And we talked about why that was important before. For the carbon reactions in the Calvin cycle, that's all taking place out here in the stroma. And so let's just, remember that for now. We're going to come back to that in just a second and kind of why that location is important. Okay, so here is just a general diagram of the Calvin cycle. And this is from your book, so if you want to look at this later, you can go to your book and get this. And you can see on the outside, it's listed kind of 5 general steps. I'll kind of walk through these. But then we divide the Calvin cycle into 3 general steps: carbon fixation, the triose phosphate production, and the regeneration of ribulose bisphosphate. And so I'll just talk about those general things. Remember, there's lots of other molecules and enzymes involved in this process, but that's more of a plant physiological kind of concept and things you would need if you're thinking more detailed about the Calvin cycle. For us, in thinking about ecophysiological responses of plants, this is enough detail for us. So the carbon fixation, let's just talk about that briefly. that is, so there's an enzyme called Ribisco. You've probably heard of Ribisco before. It's an enzyme that takes ribulose bisphosphate, which is this RUBP here, and combines it with a CO2 molecule. So it's like Ribisco is grabbing these two things and putting them together, producing PGA, which is just another molecule. We don't need to know the details of it. So that's the very first step, and that's the carbon fixation step. So we talk, sometimes when we're talking about photosynthesis, we might say carbon fixation or carbon assimilation. Those are all talking about this process of bringing CO2 molecules into, molecules in the plant and incorporating that carbon into the plant tissue. So that's that first general step. The next step is where we are actually producing some product from the carbon cycle. And ultimately, the products we're producing are simple sugars. Glucose, sucrose, which is slightly larger than glucose. Fructose is another simple sugar. So those simple sugars are being produced and is really the product I want you to remember from the carbon cycle. A couple important things here. Notice that we've got inputs of ATP. So remember, we've got ATP coming out of the light reactions, and those feed into the Calvin cycle. And then just note here, we'll come back and talk about this, but once ATP kind of releases its energy, it's broken down into ADP and inorganic phosphorus. And those will be important here, and I'll talk about them briefly, and then there's a section of the book that those become important as well. And then also we've got inputs of NADPH in this process, resulting in NADP, and that H is that hydrogen ion is contributed to the molecule and production of these simple sugars. The product that comes right out of the Calvin cycle is a triose phosphate, but that then immediately goes into those get put together into a glucose molecule. And so as I mentioned before, what I want you to remember as far as a product of the Calvin cycle is these simple sugars, or are these simple sugars, all right? So after we get a product from the Calvin cycle, then the rest of this process is just regenerating ribulose bisphosphate, which again is that molecule that gets combined with CO2 by Ribisco. There's some more energy input into these steps to regenerate ribulose bisphosphate. And it actually turns out most of the reactions in the Calvin cycle are just regenerating that ribulose bisphosphate so it can go back into that cycle. Most of the energy is spent in producing those simple sugars, but most of the actual chemical reactions are involved in that regeneration step. Okay, so that's generally what the Calvin cycle is. I just want you to remember those 3 steps of the Calvin cycle. The fact that it's producing simple sugars that can be used for storing energy long term or being used to build new tissue. And remembering that this process has a lot of energy inputs into it. And it's using the products from the light reactions to drive these carbon reactions. And then we're going to go on and talk about Ribisco just a little bit more. And remember, Ribisco is that enzyme that's combining ribulose bisphosphate with a CO2 molecule. All right, so one other thing coming back to the location that is important. So in this diagram, we looked at this last week with the light reactions, but notice that this upper side here is the stroma. So this is where those all the enzymes and molecules being used in the Calvin cycle are located. And, we draw this nice Calvin cycle, but, all the molecules aren't in this nice little circle handing things off to each other. They're all just floating around in the stroma. So all of these products that are involved in the Calvin cycle are floating out there so that the enzymes can grab them and use them and help move them along the process. The location of the Calvin cycle is important because if you notice, the production of NADPH and ATP are happening as part of the light reactions, but those products are being produced on the stroma side of the membrane. And so what that means is those products that are storing that energy are right there in the same place where the Calvin cycle is happening. So these light reactions feed directly into the Calvin cycle, and the Calvin cycle can grab these products really quick. Similarly, once ATP is broken down into ADP and inorganic phosphorus and NADP, they're also right there for the light reactions to reuse and then produce these products that are storing the energy. So it's important that where those are located, because they quickly can share the products. Also, what's important is they can quote unquote communicate with each other, right? And so, for example, if you notice here, ADP and PI, or the inorganic phosphorus, are two of the required substrates to produce ATP. If the Calvin cycle isn't using ATP, it's not breaking it into ADP and inorganic phosphorus, meaning there's not substrates for this reaction to produce ATP. If there's no, and what the important one is this inorganic phosphorus. That's a really big kind of signal atom inside the plant. So if there's no inorganic phosphorus floating around, that actually triggers the light reactions to kind of shut down. It tells that system, like, we've got plenty of energy to drive our reactions. We don't need you to work anymore, so to speak. Similarly, if there's not enough ATP coming out, like if light conditions are really low and there's not a lot of energy driving the light reactions and there's not a lot of ATP, that's going to slow down the Calvin cycle and its ability to assimilate new carbon. So there's some crosstalk between the carbon reactions and the light reactions. And so I just wanted to highlight that here because the book will talk a little bit about that. And that what's becomes more interesting from an ecological standpoint is these feedbacks between these different processes of photosynthesis. Okay, let's, summary of Calvin cycle. So just a few important things to remember. The first one is the amount of energy that's going in to the Calvin cycle to create one molecule of glucose. So we've got a lot of inputs of ATP and NADPH to produce a glucose molecule. Most of the energy, I just read energy and said it when I wasn't supposed to. Most of the reactions in the Calvin cycle are regenerating ribulose bisphosphate, but not most of the energy. And then remember the Calvin cycle is producing that triose phosphate, but then that goes to produce glucose, sucrose, fructose, these other simple sugars. They're more complex than the triose phosphate, but they're still relatively simple molecules. And then those feedbacks between the light reactions. So just remember that and how that feedback is happening. It's kind of using the NADPH and ATP as the signaling between those two processes. All right, now let's talk about Rubisco. All right, so Rubisco, the full name of Rubisco, I probably should have written this out, is ribulose bisphosphate, which is based on that molecule name. And then the CO at the end stands for carboxylase oxygenase. And we'll come back in talking about those reactions and why that's important. But first, I just wanted to put in context like the importance of this particular enzyme. So first of all, I think I said this maybe last week, but if not, an enzyme is a special type of protein. And enzymes are speeding up chemical reactions. So they're making them happen much faster than they would naturally. Rubisco is the most abundant protein on Earth. So there's approximately 0.7 gigatons, you can see there in the parentheses, of Rubisco on Earth. There was a paper fairly recently that, I was going to get my pen, in 2019, that just quantified how much Rubisco was actually on Earth. And I can imagine this was like a lab activity, right? They started talking about it in a lab meeting and they're like, I wonder how much Rubisco is actually on Earth. And so they actually did the calculation and got it into a really high impact journal, which is kind of cool. But in that paper, I forget if they gave this exact number or I converted it to this number, something that was like a little more visual. If you were to fill up 7,000 like aircraft carriers, fully loaded aircraft carriers, that's how much Rubisco would weigh. And Yeah, I don't know how many aircraft carriers are in the U.S. fleet. I guess that would be an interesting thing, but it's hard to imagine we'd have 7,000 of them. But anyway, that's a lot of rubisco, right? It's also 60% of the protein. Sorry, I'm using this drawing tablet and I'm not perfect at it. 60% of the protein in the leaves is rubisco, so it's most of what's going into the leaf. And remember, nitrogen is a really important component of proteins and enzymes in general. And so that means about 60%-ish of the nitrogen in the leaf is going just to this one enzyme. But that one enzyme has a really important job, right? Like it's that first step in getting CO2 into the leaf, that first carbon step of photosynthesis. Rubisco itself fixes 10 to 11 tons to the power of 11 tons of CO2 each year. I'll let you go ahead and calculate how many aircraft carriers that might be. But what's interesting about rubisco, we're going to talk about two problems with rubisco. And one of them is that it's actually a really slow enzyme as far as enzymes go. So it can do 3 reactions per second. And that's pretty fast for things like we can do per second, but for an enzyme in these chemical reactions, it's actually relatively slow. And that's part of the reason plants need so much of the rubisco, is to actually get the photosynthesis done at a reasonable rate. It's got a slow enzyme, and so it needs lots of it. All right, let's talk a little more about the downsides of rubisco. So good, I did put the full name here of the enzyme. And I'm not really expecting you to know the full name, but remember rubisco, and then I do want you to remember what the C and O stands for. So that stands for carboxylase and oxygenase. So rubisco can perform 2 types of reactions. When it performs a carboxylase, that's what we normally think of rubisco doing. That's when it takes ribulose bisphosphate and a CO2 molecule and combines those. That's a carboxylase reaction or carboxylation. Oxygenase is when it takes the ribulose bisphosphate and combines it with an oxygen molecule. And of course, that results in a different product and kind of a different pathway. And it turns out that reaction happens about 20% of the time. So about 1 in 5 reactions, rubisco accidentally, if you will, takes an oxygen molecule instead of a CO2 molecule. And this process results in a loss of CO2 from the leaf. So instead of gaining a CO2 molecule, it's actually losing a CO2 molecule. And because it's releasing CO2, which is a respiratory process, this process, when rubisco reacts with oxygen, is referred to as photorespiration. All right, so photorespiration is when rubisco reacts with oxygen and releases CO2. It also produces a product that is toxic to the leaf, and under normal situations, it's not a problem. That it can take care of that toxic compound. But if photorespiration starts happening very frequently, then that can actually cause a problem and start shutting down other physiological processes. So let's look at that briefly. Let's see. There we go. So here we have on the y-axis we have carbon assimilation. On the x-axis we just have time, and then this is just a representation of what photosynthesis looks like under different concentrations of oxygen. So in this case, what the study did is put air into a chamber around a plant. And the air it was putting into that chamber had different concentrations of oxygen. Currently in our atmosphere, we're around 20% oxygen. So the air around us is about 20%. So then they doubled it and did 40%. And then we also have 0.5%. And this is the concentration of oxygen they think the atmosphere was at when rubisco evolved. So what you can see is if you, under really low oxygen conditions, photosynthetic rates are going to achieve a higher rate than if you have really high concentrations of oxygen. And that's due to photorespiration. So if you don't have very much oxygen around, then Rubisco is not going to accidentally react. I'm going to put quotes around accidentally. It's not going to react periodically with oxygen, right? It's going to react with CO2 more often because there's just no oxygen around. But if you start putting more and more oxygen into the atmosphere, that oxygen gets into the leaf where Rubisco is, and then Rubisco starts reacting with oxygen more frequently. And that's why photosynthesis goes down, is because it's respiring more. That's especially important when we think about plants growing in warm environments. So, enzymes have a specific structure to them, which allows them to kind of fit with particular molecules. So it's almost like a lock and key kind of thing. Certain molecules will fit into a space within the enzyme, and that's what helps these enzymes be specific to different reactions. So under, cooler temperatures, let's say around 20 degrees, Rubisco fits best with CO2, and that's why it reacts with it more frequently. But, oxygen can kind of fit in there, and so it does react with oxygen once in a while. As you warm up this enzyme, though, it changes the structure enough that it reacts more frequently with oxygen. And so that's why we see this exponential curve here. So on the x-axis, we have air temperature. On the y-axis, we have the relative ratio of photorespiration to photosynthesis. So, and we're going to be looking at the solid line. You can see as you increase temperature, the relative ratio increases, which means the proportion of photorespiration is increasing at an exponential rate. So the warmer and warmer it gets, the more and more photorespiration is occurring. That means the plant is not fixing as much CO2, it's losing CO2 through the photorespiration process. And the chance of those toxic compounds building up in the leaf increases as well. And so this is one of the reasons why really high temperatures really impacts the growth of plants, is because it's reducing the efficiency of plants taking up CO2. Okay, and then just a couple summary things on Rubisco. Again, Rubisco's using oxygen instead of CO2 when it's photorespiring. That sensitivity changes with temperature. And we think there's good evidence that Rubisco evolved during times when, or during a time when atmospheric CO2 was really low. And so it wasn't a problem. I mean, we talk about now about these silly things Rubisco does, right? Slow reactions and the fact that it reacts with oxygen and causes these problems for a plant. But when it evolved, like, oxygen wasn't a problem, so it wasn't a big deal. And we know that evolution isn't necessarily a conscious process, so even though I kind of talk about it that way. And the energy that's used for those reactions, those processes are still using ATP and NADPH, but then of course they're not going to carbon gain, they're going to carbon loss. The one thing I do want to point out that it's not all negative. Photorespiration can help dissipate excess light. So let's say for a second we've got full sunlight, so we have lots of energy coming into the leaf. But let's say for some reason the plant doesn't have enough CO2. And this can often happen when plants are closing their stomates to conserve water. So we end up with a situation where the carbon reactions aren't necessarily using the ATP and NADPH. So remember back to when I was talking about the feedbacks, if the carbon reactions aren't using that energy, then what happens is that shuts down the light reactions. If the light reactions are shut down and there's still a lot of radiation hitting the leaf, that's going to heat up the leaf a lot more, because one of the fates of that energy coming into the leaf is producing heat. So if the carbon reactions aren't happening when it's full sunlight, then that leaf is going to heat up and probably experience a lot of damage. So in a situation where the leaf doesn't have a lot of CO2 to drive those carbon reactions, it's actually helpful for the plant to photorespire, because then it's using ATP and NADPH. It's telling the light reactions that they can keep working, and so then the leaf can use that energy to drive these reactions versus it just heating up the leaf. So there's some situations where photorespiration can benefit the plant and help it kind of tolerate these really warm temperatures. All right, so as I did last week, try to just highlight some of the terms and concepts I want you to remember, because then we'll be reading some papers that will be incorporating these terms and concepts, and it'll just make that reading a lot easier. So remember the Calvin cycle, the 3 general steps that are in the Calvin cycle, and the products that are associated with that. Rubisco, what is Rubisco? Why is that name important? And remember the problems with Rubisco. And then just remembering how ATP and NADPH in particular are kind of being used for communication between carbon reactions and the light reactions. And then just know a little bit about photorespiration is, what that process is, and why it's a problem, but also why it can be an advantage under some situations. And then just to try to relate these more to an ecophysiological standpoint and where we would see variability in these processes. Certainly we see huge variability in the amount of Rubisco in leaves. There's just interspecific differences between species and how much Rubisco they have for various reasons. So we see that can be a difference between species in the rate of photosynthesis they can perform. Soil fertility, talked about how nitrogen is really important to how much Rubisco a plant can have. So certainly under low nitrogen conditions, that's one of the biggest impacts on the plant, is that it might not be able to produce as much Rubisco, and that's going to affect its photosynthetic rates. And then certainly light conditions are going to affect the amount of Rubisco. Because nitrogen is such an important part and it's a relatively hard thing for a plant to get. If you're in a low light environment, if you're a plant and you're in a low light environment, you don't want to invest a lot of your nitrogen into Rubisco, because there's not going to be enough energy to drive the processes that Rubisco performs. And so the light environment plants are growing at impacts how much Rubisco they're going to have in their leaf. The substrate supply for the Calvin cycle, how fast CO2 is getting to Rubisco and these reactions is going to impact how fast they're happening. And that's something that's really changes drastically over the course of the day and the season. And I'll talk a little more about that in the next video, but just want to highlight that here. We talked about that communication between Calvin cycle and light reactions, and so the supply of energy is going to impact how fast these reactions are happening. And then photorespiration, there's variability between species in their rates of photorespiration. Their sensitivity to temperature differs between them, and part of that is due to photosynthetic pathways, so C4 versus C3 plants. We'll talk about that more in the coming weeks, but that's certainly another difference between species that we see. How much photorespiration changes over the course of the season, I don't actually know. That just popped in my head. That it's not something I've read and noticed in the literature, but I wouldn't be surprised if it's out there. So if we get interested in that more, we could look that up in the literature and see what's available. Okay, that is it for carbon reactions. I'm going to stop here. Let's go back here. And then I'll do another video on substrate supply.